Bacterial surface proteins make direct contact with host cells and are also involved in uptake of nutrients from the environment. Therefore, surface localization of bacterial proteins is a key step towards identification of virulence factors. Here, a method to assess surface exposure of proteins on intact leptospires is presented. In comparison to previously described immunofluorescence assays, the details of the method described here are designed to minimize the manipulation of cells in an effort to maintain outer membrane integrity. First, leptospiral suspension is added to glass chamber slides. The slides are incubated to allow the cells to adhere. Adherent leptospires are fixed with 2% paraformaldehyde to preserve an intact outer membrane for surface protein assessment, or with 100% methanol to permeabilize the outer membrane to reveal subsurface proteins. The fixed leptospires are then labeled with specific antibodies and visualized by fluorescence-labeled secondary antibodies. Surface-localized proteins can be labeled and observed in intact and permeabilized cells, while subsurface proteins can only be observed in permeabilized cells.
This method can help answer key questions in the microbial pathogenesis field, such as, is a protein localized on the bacterial surface, where it can play a role in host pathogen interactions?
The method presented here is designed to minimize the manipulation of cells, which should help to maintain outer membrane integrity. This method involves a lower concentration of fixing agent, washing with outer membrane stabilizing buffer, and fewer washing steps than previously described assays.